What's going on, everybody? Welcome to The Curve, the channel where we look at health trends on social media. Today's trend is going to be GLP-1, specifically Ozempic. Ozempic, as we know, is one of the most talked about drugs in the world right now. It's on the news, social media, conversations at the gym, the doctor's office. We see celebrities are using it to lose weight. Politicians are debating whether it's good or not. And we see pharmaceutical companies are profiting billions from this one drug. But here's where I've noticed this week covering this space is that almost nobody, including most people taking these drugs, actually understands the biology behind them. And that matters because when you understand the mechanism, everything else makes sense. For example, why it causes weight loss, why it affects your appetite, why it has certain side effects, and why newer drugs in the same class are producing results that would have seemed impossible 5 years ago. We're seeing this with retatrutide, which is a triple agonist, and we're seeing the incredible effects it's having and how it's trending all over social media. But we'll cover retatrutide in a different episode. And the thing that surprised me most when I went on with the research about GLP-1 is that most people on these drugs are spontaneously stopping drinking, losing interest in gambling, and they're reporting changes in behavior that have nothing to do with weight loss. And the last point about addiction is that it comes from a 2022 paper in the British Journal of Pharmacology, and we're going to get into that later on in the video. But first, let's start from the beginning so it all makes sense. So GLP-1 stands for glucagon-like peptide. It's a hormone your body produces naturally every single time you eat, and it belongs to a class of hormones called incretins, which are gut-derived signals that tell the rest of your body that food has arrived and it's time to start managing it. So here's where it gets interesting and where most explanations stop short. GLP-1 doesn't just appear out of nowhere. It's produced through the breakdown of a larger precursor protein called proglucagon. And according to a 2019 review in neurochemistry from researchers at Vanderbilt and the University of Alabama, proglucagon is expressed in 3 distinct locations in the body. It's produced in your pancreatic alpha cells, an enzyme called PC2, for hormone convertase 2, breaks proglucagon down and produces glucagon, which is a hormone that raises blood sugar. And then in your intestinal L cells and in the brainstem, a different enzyme called PC1 and PC3 breaks the same starting protein down but produces GLP-1. And what we're seeing is that the same gene and same precursor protein has a completely different hormone outcome depending on which tissue is in the processing. Which brings us to where GLP-1 is actually made. And this is the part most coverage of this drug class gets wrong. So first, we have the L cells of the small intestine. This is your primary source of circulating GLP-1. The density of these cells actually increases as you move further down the gut. For example, there's more cells in the ileum than the duodenum. And second, we see that the alpha cells of the pancreas also produce this, which is a secondary source. It processes the same proglucagon protein, but produces primarily glucagon with smaller amounts of GLP-1 compared to the gut. And then third, this is the one that changes the story entirely, is the nucleus of the solitary tract in your brainstem, the NTS, also produces GLP-1. And this is really fascinating because the NTS is a major relay hub for visceral sensory signals, which is essentially hunger, satiety, nausea, and cardiovascular information. And the fact that GLP-1 is manufactured there directly shows us that GLP-1 is not just a gut hormone that eventually drifts up to the brain. It's actually produced by the brain itself. Now, here's the main problem, is that natural GLP-1 has a half-life of approximately 1 or 2 minutes in circulation, and 2 mechanisms destroy it almost immediately. An enzyme called DPP4, which is dipeptidyl peptidase 4, cleaves it into an inactive form and it continues to clear the remainder that's left. And 2 minutes, that's how long your body's own GLP-1 survives before it's gone. This is why the pharmaceutical development of GLP-1 drugs was such a significant scientific achievement, is because the challenge wasn't just mimicking the hormone itself, it was engineering a version that could survive in the body long enough to actually do something meaningful. And this is where we get semaglutide, which is Ozempic, and it has a half-life of approximately 7 days, which is why you only inject it once a week. So getting from 2 minutes all the way up to 7 days to decades of molecular engineering, and that gap is essentially the entire story of how this drug class was built. So how does GLP-1 actually work at the molecular level? So when GLP-1 binds its receptor, B family G protein coupled receptor, it sets off a 5-step cascade. So here we have step 1, the receptor activates adenylate cyclase, and then step 2, adenylate cyclase converts ATP into cyclic AMP, also called cAMP. Then step 3, cyclic AMP activates protein kinase A, PKA, and then step 4, PKA closes ATP-dependent potassium channels, depolarizing the cell membrane. And then step 5, which is arguably the most important step, that depolarization opens calcium channels, calcium flows in, and insulin is secreted from beta cells. But yeah, that's the critical detail, and that's what makes GLP-1 drugs fundamentally safer than older diabetes medications, is that insulin is only released when blood glucose is already elevated. So essentially, if your blood sugar is normal, GLP-1 does not trigger insulin release. That built-in glucose-dependent mechanism is why this class doesn't cause dangerous hypoglycemia. So GLP-1 doesn't just act on one system, and it acts on 3 simultaneously. In the pancreas, it triggers insulin release only when glucose is elevated, suppressing glucagon, and according to the Zhao and colleagues at Shaanxi Medical University, actually promotes beta cell proliferation and survival. That's not just managing a disease, that's potentially preserving the function that produces insulin in the first place. Then in the gut, it slows gastric emptying, which means food moves more slowly from your stomach into your small intestine, and blood glucose rises more gradually, and you feel full for longer. And that slower emptying is also a key driver to the GI side effects we'll cover later. And then in the brain, we see it reduces appetite. Signals in the hypothalamus increases satiety signals and reduces the reward value of food. So that's what we're seeing GLP-1 is acting simultaneously on. It doesn't just affect one thing in our body, but in reality, it affects 3 whole different pathways. So this is the mechanism behind something a lot of people on GLP-1 drugs describe but can't explain. They don't stop eating because they feel uncomfortably full. They stop eating because they simply just don't think about food as much and they don't crave it. The drive is reduced at the level of the brain's reward system, not just at the level of the stomach. So essentially, you don't stop eating because of willpower. You stop eating because the neurochemical signal that makes food compelling has been quieted. So essentially, there's times when you forget that you need to eat because you just don't have this feeling of hunger anymore. Now let's talk about how the brain signal actually gets there, because it's where it gets genuinely interesting. So GLP-1 secreted in the gut acts on GLP-1 receptors on the vagus nerve, exciting it. The vagus nerve then stimulates NTS neurons in the brainstem, and those NTS neurons then release their own GLP-1 directly in the brain. So here's why that matters. So circulating GLP-1 is degraded by DPP-4 before it even reaches the brain, and it can't get there through the bloodstream. The half-life is too short. Instead, the gut sends a neural signal via the vagus nerve, which then triggers GLP-1 production in the brainstem itself. This was confirmed by vagal deafferation studies when researchers cut the vagus nerve. The appetite-suppressing effects of peripheral GLP-1 were significantly reduced. And the effects don't stop at appetite and blood sugar. According to Zhao and colleagues, GLP-1 receptors are found throughout the body, and the implications are actually significant. First, we have cardiovascular, which is GLP-1 receptor agonists reduce blood pressure, improve microvascular function, reduce inflammation, and have shown reductions in major cardiovascular events in large clinical trials. And then next, we have neuroprotective. GLP-1 receptor agonists stimulate nerve cell differentiation and inhibit neuroinflammation. There's active research into applications for neurodegenerative disease, which GLP-1 may actually aid in preventing. And next we have metabolic, which activates brown adipose or fat tissue metabolism and increases energy expenditure through pathways independent of sympathetic nervous system. And then we have liver related, which inhibits inflammation and reduces fat deposition, which is relevant for non-alcoholic fatty liver disease. So we're seeing that GLP-1 not only reduces hunger, but actually beneficial for so many different things, which is why it's such a great scientific achievement. Now, here's the part that nobody's talking about, and the finding that genuinely surprised me most when I went through my research over the past week is that a 2022 review in the British Journal of Pharmacology by Clausen and colleagues at the Psychiatric Center of Copenhagen looked at the relationship between GLP-1 and addictive disorders. So GLP-1 receptors are expressed in the ventral tegmental area in the nucleus accumbens. If those names don't ring a bell, those are the core structure of the brain's dopamine reward system, which is the same circuits that drugs of abuse hijack. For example, alcohol and all these different drugs like nicotine, cocaine. But what the preclinical research found is that alcohol intake was reduced in rodents and non-human primates. A clinical trial using exenatide was conducted in alcohol use disorder patients, and cocaine's rewarding effects were unattended in animal models. Then we also have amphetamine, which induced locomotor activity was reduced, and then nicotine intake fell in preclinical models. One important distinction, and the paper is clear about this, GLP-1 receptor agonists do not appear to affect opioid abuse-related effects. And the mechanism seems specific to alcohol, stimulants, and nicotine, not opioids. The researchers are careful to note that precise mechanisms are still being uncovered. This is not an established treatment for addiction, but it is plausible biological mechanism for something a lot of people on semaglutide have reported. Spontaneous drinking less, losing interest in other compulsive behaviors, the dopamine reward circuits have been modulated, and the full implications of that are still being worked out. So this is a great advancement, not only to treat diabetes, to treat obesity, but also to help with addiction and can be used in therapeutic ways. So let's bring it back to the numbers because the clinical data is what makes this drug class impossible to ignore. So a 2023 comprehensive review of randomized controlled trials by Popoviciu and colleagues synthesized the evidence across the full GLP-1 drug class. So placebo, roughly 2% body weight lost. And then we have liraglutide, the first GLP-1 agonist approved for long-term weight management, was around 7%. Semaglutide at the 2.4 milligram dose was approximately 15% over 6 to 8 weeks. And then tirzepatide at 15 milligrams was approximately 22%. So we're saying that all of these also showed improvements in hyperglycemia, which is elevated blood sugar, insulin sensitivity, blood pressure, and cardiometabolic markers, as well as renal kidney function. So the most important part is the side effects, because we have to discuss the downsides as much as we want to discuss the upsides. So the most common side effects, which aren't as major, are nausea, constipation, vomiting, gastrointestinal discomfort, primarily during dose escalation. And these are real side effects, but most manageable, and they're not that uh, filtered risk. And they improve substantially as the body adapts slowly to the GLP-1. And then next we have the more serious considerations, which is pancreatitis risk, which is an important caveat here. Multiple meta-analyses found no significant increase in pancreatitis incidence with GLP-1 receptor agonist use, and obesity itself is confounding risk factor for pancreatitis. You have thyroid C cell findings, which was observed in animal studies, which is why these drugs are contraindicated in people with a personal or family history of medullary thyroid cancer. So yeah, nothing in this video is medical advice. These are real considerations that belong in a conversation with your physician. But where it's heading. So the progression of this drug class follows a clear biological logic that we're seeing is that GLP-1 receptor agonism has a powerful incretin effect, approximately 15% weight loss. Then add GIP receptor agonism and synergistic effects on insulin and fat metabolism. There's trizipatide, which is approximately 22%. And then if you add a third, glucagon receptor agonism, elevated energy expenditure, the metabolic rate goes up. Then we have retatrutide, which is trending all over social media now, and is approximately a 24% body weight decrease. And a 2022 paper in Cell Metabolism from Eli Lilly's own research team showed that in obese mice, the triple agonists produce greater body weight loss than trizepatide alone, associated with increased energy expenditure beyond what appetite suppression alone would explain. And phase 3 clinical trial data confirmed those results in humans. And we're going to do a full dedicated episode on retatrutide, like I said, soon, the mechanism, the trial, the data, and what it means for the future of metabolic medicine. But for now, we're just focusing on GLP-1. So in summary, GLP-1 is not a weight loss hack. It is a hormone that evolved as part of your body's system for managing energy. And the drugs that target it are extended and amplifying the system that already exists. It's produced from proglucagon in 3 locations: the gut, the pancreas, and the brainstem. It's processed differently by different enzymes in each tissue. It acts on the pancreas, the gut, and the brain simultaneously. The brain connection is real, mediated by the vagus nerve and the NTS neurons manufacturing their own GLP-1. And early evidence links the same receptor system to dopamine reward circuits, including addiction pathways. So here are the references. All sources are going to be linked in the description below if you want to look at them yourself. It's pretty interesting stuff, and the papers are freely available on PubMed Central. So that's what this channel's here to do. I'm not hyping any drug up. The quick answers, the real biology, where it comes from, what the evidence says, what it means, side effects. We're keeping it real. There's new videos every Wednesday. Next week, the episode's going to be the top peptides of 2025. The ones with real data behind them. So probably expect retatrutide, BPC-157, MK-677, GHKCU, things like that. And then we're also going to cover the ones that aren't worth your attention. So subscribe so you don't miss it. And then I'll see you guys then.